Yeah, good morning. My name is Ibne Ali, and I'm from CDC. So I have nothing to declare in terms of financial disclosure. And so this is the outline of my talk. Today, I'll talk about some tools that are available for Naegleria fowleri detection. And then I'll talk about the laboratory testing that we performed on the Sebastian case. And I'll talk about how we can improve PAM diagnosis. And finally, I'll briefly talk about our whole genome approach to develop new genotyping tools. So these are the tools that are currently available for diagnosis of Naegleria fowleri. These include microscopy, real-time PCR, indirect immunofluorescence assay to detect either intact amoeba or antibodies. So before I talk about microscopy, I should mention that Naegleria fowleri has three life stages. One is trophozoite, the invasive form, motile form. The swimming state is flagellum, and the highly resistant environmental form is the cyst. But it is only the trophozoite form that is ever found in the brain of the patient or in the CSF. And as a diagnostic technique, microscopy is really good because it would be fast, it would be cheap, it will not require any sophisticated instrument. But it has several limitations too. It will need expertise to know the amoebic morphology, and it has low sensitivity, it is not specific. Oftentimes, white blood cell leukocytes are misdiagnosed as amoeba, and you need to see intact amoeba or mortality in order to diagnose by microscopy. So our recommendation is, even if a specimen is positive by microscopy, it should be verified by molecular assays, such as real-time PCR. So at CDC, we have a real-time PCR that is based on mitochondrial DNA, and there are several thousand copies of mitochondria per cell in Naegleria fowleri. As a result, this PCR is extremely sensitive. It can detect even one amoeba, showing here. And the assay completion time is only 2 to 3 hours, including the DNA extraction step. But main limitations are it will require expertise, and it will also require sophisticated and expensive instrument to do the testing. And we also have a serological test to detect amoeba in serum, and the titer of greater than 1:164 is considered positive. It is species specific, and assay completion time is 4 hours, but it has two major limitations. A negative test result will not rule out an active infection because antibody response require at least few weeks to develop. As in most PAM cases, we do not see an antibody response. And even if a serology is positive, it will not differentiate between a past exposure and present infection. That's why serology is not very good in terms of diagnostic technique. So we also have an indirect immunofluorescence assay to detect intact amoeba, but it requires brain tissue. As a result, it's mostly done on the autopsy cases. And sensitivity is not as high as PCR, but it is species specific. It takes about 24 hours. As a result, it's laborious and time-consuming, needs expertise on morphology and immunofluorescence, and definitely collection of brain tissue is a very invasive procedure. Now I'll talk about the Sebastian case, what we did in terms of testing at CDC. So this is the timeline. We first learned about this case on 8/7 on Sunday. On Monday, Florida Hospital arranged to send the specimens to CDC. And these are all CSF specimens. On Tuesday, we received 2 specimens, and both of them were positive by PCR. And on Wednesday, we got another one. That one was also positive. And the next 2 were negative, which was very good. We were very happy to see that. And Florida Hospital kept collecting specimen Friday through Monday, 4 samples. And we received another batch of samples on Tuesday. But surprisingly, the Monday, Friday, and Saturday specimen came back weak positive. And we repeated these samples, and they were genuine positive. But the next 2 were negative. In fact, the remaining 3 were also negative. And so this is the summary of the PCR. So first 3 were positive, then 2 were negative, and then again 2 came back positive, and then remaining 5 were negative. And at that point, they stopped collecting CSF. So in addition to CSF, we also received 6 serum samples. 5 of them were collected daily from 8/11 through 8/15, and 1 was collected when he was released on 8/23. And first 5 were actually negative because the titer was less than 1:164. But there was a trend that it would be positive. And in fact, the last one was positive, 1:128. And at 1:128, we took the IFA pictures, and this is a positive control, which was staining the coated amoeba. And this is the first serum, day 1 serum, which was not staining any amoeba. And this is the last serum, which was clearly staining amoeba. So in addition to doing PCR with the CSF, can you please run this video? This is a video. Yeah, so in addition to doing PCR, we put all the CSF in culture, and only the first CSF was positive. You see this movement. And this is surprising that we only saw 1 amoeba on Friday after inoculating it on Tuesday, and the number never got very high. The maximum I saw was about 10 amoeba, and they are not even growing very well. So we are trying to keep it in the lab, but still we haven't been very successful. So some preliminary conclusion based on this patient is that the consecutive CSF collections were very helpful for monitoring amoebic DNA. And if the patient is recovering, based on this case, I think we should at least see 3 consecutive CSF as negative before we can say that the patient is free from amoeba. On IFA serology, we found that it took about 3 weeks to develop a positive antibody response, and we'd like to monitor this patient to see how long the antibody persists. And on culture, it looks like the amoeba strain was weak. It most likely not the case of drug. I heard from Dr. Jordan that the drug treatment was initiated after the first CSF collection. So it's not likely the drug, but it may be possible the strain was less virulent or the inoculation dose was low. That's why it's not surviving in the culture. So now I'll talk about how we can improve PAM diagnosis. So one way we can improve the PAM diagnosis is by training the pathologists or laboratory technicians so that they can become familiar with Naegleria fowleri morphology and mortality. As I mentioned, leukocytes are often misdiagnosed for Naegleria fowleri. And we can also aim for developing improved tools which would be simple, no expertise or sophisticated instrument will be needed, and one that can be performed at patient's point of care, which will be rapid, sensitive, and specific. And one such test will be an enzyme-linked immunosorbent assay, or ELISA, and it will definitely target amoeba antigen in the CSF. And in fact, there are 2 or 3 ELISA that have been reported in the literature, but they have never been tested in actual clinical specimens. And at CDC, we are trying to develop an ELISA using 3 monoclonal antibodies. Yeah. And other improved tool can be isothermal amplification, which will be alternate to normal PCR, conventional or real-time PCR. The major advantages would be that we should be able to do it at a constant temperature without needing a PCR instrument. And it can be done using a water bath, heat block, something like that, or even in the room temperature. And so it can be performed in the non-laboratory settings. And because the test is based on nucleic acid, it will not require intact amoeba structure. And the result can be read visually. And one such isothermal amplification is loop-mediated isothermal amplification assay, or LAMP assay. And one has been developed for Naegleria fowleri. And the target is DNA, specimen is CSF. The specimen would be CSF, but the one that was developed, that has never been used in clinical patients, specimen. So we want to test it for clinical patient and see how it works. And it should be species-specific, and it would be rapid. So now I'll talk a little bit about whole genome sequencing that we are doing at CDC in order to develop novel genotyping tools. And before going into genome sequencing data, I want to show an electron microscopy partial picture of Naegleria. And this is the nucleus, and these are the 4 mitochondria. And both nucleus and mitochondria have their own genomes. The mitochondrial genome, which is circular, is about 50 kb, encodes 48 genes, and it has several hundred to thousands copies per genome. And the existing genotyping is based on this mitochondrial genome, mitochondrial DNA. And nuclear genome is much bigger, 29 megabases. It encodes approximately 13,000 genes, and no genotyping tools are available based on nuclear genome. So in CDC, we are trying to understand why, although PAM, Naegleria fowleri exposures appear to be common, but why the cases are not so common. So we think it's linked to the inoculation dose of the amoeba, or host susceptibility, or virulence of the infecting strain. But existing genotyping system categorize all the US and Naegleria fowleri into only 3 broad genotypes. As a result, it does not show any links to amoebic virulence, and it's not very useful in molecular surveillance. So in this whole genome sequencing, we have aimed to develop a high-resolution strain typing or genotyping system based on single nucleotide polymorphism, or SNPs, and repetitive DNAs. We also want to identify virulence genes and genes whose expressions are linked to virulence, but I'll not go into any details on these 2. So I'll talk about single nucleotide polymorphism. So single nucleotide polymorphism, or SNP, is a variation in a single nucleotide position that occurs at a specific position in the genome. So these are the DNA sequence from 9 strains, and I highlighted 3 areas where we see nucleotide SNPs, like here A, here T, something like that. And in mitochondrial genome, between the 2 different genotypes, there are about 600 SNPs between any 2 different strains. if they are coming from two different genotypes. But within the particular genotype, like genotype 1 strain, there were about 0 to 60 SNPs. But in nuclear genome, there are about 50,000 SNPs between any two strains. So this is the table showing some of the SNPs that we detected in different strains in genotype 1 strains. So these are the three I highlighted here, three mutually exclusive SNPs that we can utilize to develop new genotypes. And we try to use these SNPs in order to identify the source of infection using 4 PAM cases and related environmental samples. And when we used mitochondrial SNPs, we found only 1, 2, or no SNPs in these 2 cases between PAM strain and environmental strain. So we thought we've identified the strain that was responsible for PAM. But when we looked at the nuclear genome, there were like 45 or 47 different, 47,000 SNPs. So the strains were not identical. So our 2 major conclusions are that mitochondria, we should not use mitochondrial genome for infection source tracking. And it looks like, because we never identified the actual strain that was responsible for PAM, we think the strain that is responsible for PAM is a rare or minor population, and that are like more virulent. That's why they are causing the disease. And we are also trying to use repetitive DNA sequence, and this is an example of repetitive DNA sequence in several isolates. So this is a like 15 base pair repeats that has been repeated 2 times in these 2 strains, 1 time in these 3, but it's completely missing in these 3. So based on repetitive DNA sequence, we are trying to develop a new genotype. And this would be better than SNP because we should be able to see the difference just by running the PCR product into gel. We will not need to sequence the amplified product. So I think that was my last slide. So overall take-home messages are, all the current diagnostic tests are based on laboratory settings. So we want a tool that we should be able to perform at patient's point of care, which will be simple, rapid, and highly sensitive. And we believe powerful new genomic technologies and genome sequencing will help us to develop new improved genotyping tools, improved diagnostic tools, and help us understand the virulence. And regarding the 4th survivor case, in addition to saving Sebastian's life, the Florida hospital has done a very good, excellent job by doing a comprehensive sample collection. I'm sure this will prove important in the future. So with this, I would like to thank the organizers of this Amoeba Summits and Mr. and Mrs. Smelski. And from Florida Lab, I would like to thank Dr. Laham, Dr. Jordan, and Brandarak for providing us with the samples and collaborating with this Sebastian case. And from CDC, I would like to thank Dr. Jennifer Cobb because she introduced me to this summit. And Shantanu Roy and Alicia Kelly is from my lab. And also, I would like to thank Vincent Hill and Katie Fullerton. And thank you. Yes.
I want to publicly acknowledge Dr. Ali's work. He was of tremendous help. I know for him, I was sending candy to his lab, but for us, he was able to process specimens immediately. And he was in permanent contact. Sending stuff to the CDC is not an easy task. There's a lot of moving parts, and we were able to kind of breeze through that. So I want to
thank you.
One last thing is that probably will come up out of the sessions of lunch, but the question to you will be, if we were to choose one technique, do you think that microscopy alone is enough for a hospital, or should we be thinking about more sensitive methods?
Yeah. If we have highly trained pathologists or technicians, then microscopy alone would be very helpful. But eventually, I think we need to go towards molecular testing, simple and rapid molecular testing. Yeah.